Hello, I'm Richard Young, your host with Facundo Batista and Lena Fayan for MIT's course on COVID-19, SARS-CoV-2, and the pandemic. There are well over 200 virus species that are known to infect humans, and several new species are discovered each year. At least 2/3 of these also infect non-human hosts, and some of these have an ability to jump from one host species to another. This cross-species transmission from animals to human depends on many factors: exposure to the virus, the presence of an adequate receptor on the surface of our cells, the ability of the virus to exploit our own cellular machinery to replicate and escape our many defenses, and the ability to be transmitted from one person to another well enough to cause epidemic outbreaks. The purpose of this course is to learn what we know today about COVID-19 from the world's top scientists. Last week, Dr. Bruce Walker introduced us to the broad scientific issues surrounding the pandemic. Today, I have the pleasure of introducing you to a talented virologist, Dr. Britt Glaunsinger, who has been at the forefront of educating both scientists and the public about the novel coronavirus. Dr. Glaunsinger is a professor at the University of California, Berkeley, and a Howard Hughes Medical Institute investigator. Her award-winning research is focused on the ways that RNA viruses hijack the machinery of host cells. Professor Glaunsinger, thank you for bringing our current understanding of this virus to our students and viewers.
Thank you so much for the invitation. Just wanting to make sure you can see my screen now before I get started. Yeah, okay. So I'm really delighted to be able to participate in what is looking to be a pretty exciting class this semester. As Rich mentioned, for most of my career, I've studied how viruses manipulate host gene expression and co-opt the cellular gene expression machinery in order to benefit the virus and its replication cycle. Actually, I've generally studied this during DNA virus infection, but since March of this year, of course, this has expanded to include coronaviruses. And as Dr. Walker pointed out in his seminar last week, it's really part of an unprecedented worldwide shift in the focus of scientists of all disciplines towards the singular goal of understanding this new virus in order to fight the pandemic. And I just want to take a second to highlight that, in fact, many of these efforts are volunteer-based and should be acknowledged. And so I just want to mention that in this slide, for example, these are two of the postdoctoral fellows in my lab, Azra Lari and Divya Nandakumar, who over the last many months have donated a lot of hours of their time to help set up and run a COVID-19 diagnostic testing lab that was organized at UC Berkeley through the Innovative Genomics Institute, both for testing campus and also for testing underserved components of the community where this has been limited. And so although these are bench scientists, they've gotten into the clinical realm just in order to contribute to these pandemic efforts. And I really liked Azra's statement here, which was that I initially went in tepid about how much I could offer, but there really isn't an insignificant part of the process. Every seemingly small task contributes to the larger goal. And to me, this is really a reminder of the power of working in a coordinated and a collaborative way, which we're seeing a lot of during this pandemic. It may be one ray of light in what is otherwise a lot of darkness here. So the part of this broader story that I'm going to tell you about today is how the virus itself operates. And that's because the better we can understand how the virus works, the better prepared we are to design really effective interventions. And I'm going to break the seminar into three general themes. Today, we're going to begin discussing in some detail how CoV-2, which is the name of the virus that causes COVID-19, of course, gets into your cells. And then once it's there, how does it activate the viral machinery that is encoded in its set of genetic instructions, its genome? And then finally, how does it use that machinery to dramatically reorganize the cell and convert it into a virus-making factory? I know that we've got a pretty diverse audience in terms of academic background listening in today. So what I'm going to do is provide certainly some molecular details on each of these topics. But overall, I'm going to try to keep most of the lecture at a level that I think a freshman level science student could understand. So if you start to get lost, don't worry. I'm going to try and zoom back out to the bigger picture pretty regularly. All right, let's begin by just going over what makes up a coronavirus particle. Actually, what makes up any virus? The definition of a virus is basically something that has a set of genomic instructions, genetic instructions. It can be either on DNA or on RNA. For coronaviruses, it's on RNA. And that set of genetic instructions is protected in a house that is made of either protein or a mixture of protein and lipid or fat, which is called an envelope. And the proteins come from the virus, but anytime there's a lipid involved in a viral particle, that's always stolen from the host. No virus can make or generate its own lipid molecules. So let's just take a coronavirus image and slice it in half for this diagram to look at what are the components of the coronavirus particle. You'll see in orange is the RNA genome. It's wound around the inside of the particle. It's a strikingly large genome. We're going to talk about this in more detail later. It is a genome that is what we would call positive sense RNA. That just means it's ribosome ready. Once it goes into the cell, it can be read directly by the protein machinery. And that genome itself is coded in a viral protein called nucleocapsid. This is something that both helps protect the RNA, but also is important, we think, for helping these new viral particles form after the RNA has been replicated, process of morphogenesis. And that nucleocapsid RNA is then surrounded by a lipid envelope that is, as I said, taken from the host cell. And that lipid envelope is studded with 3 different viral proteins, 2 of which, the matrix protein shown in purple, and the envelope protein shown in yellow here, are involved again in formation of the virus particles, helping the viral RNA bud into them. The third, the spike protein, is what gives the virus its name. So, and that comes from looking at this virus under electron micrographs. This spike protein, which is the most prominent feature of the viral particle, is something that stands out, looks like a corona, which means crown, a crown of spikes, or a halo in an eclipse. And the spike protein, we're going to spend some time talking about because it plays 2 critical roles for the virus in enabling it to get into cells. It is also the major target of neutralizing antibodies to inactivate the virus. And thus, the focus of almost all vaccine efforts are to generate neutralizing antibodies against this spike protein. So let's take a closer look at spike. Spike is actually a group of 3 of the same protein. It's a homotrimer that come together that form each of these structures on the surface. We actually know from a number of cryoelectron microscopy studies, we know a lot about the spike protein at atomic level detail. So I'm just showing you an image from one of those publications here. And to highlight that spike has a couple of different regions or domains that are important. First is this upper globular domain here, which is involved in receptor engagement. So you can think of spike as a key that is looking for a cellular lock to get into. It can only insert into a lock that will fit the key, and so only cells that have the right lock can be accessed by the virus. And ACE2 is the name of that lock. It's the protein or the receptor on the surface of cells that allow the virus to get in. So this upper globular domain is the part of spike that binds that receptor. But that's not the only thing that spike is doing. In fact, it's got this lower domain that houses a second critical activity. Essentially, it's got what's called a fusion machine or a fusion activity buried in this lower stalk-like region of the protein. You can think of the fusion machinery as something that's spring-loaded, like a jack-in-the-box, where when it's got the lid closed on the box, that spring is in a condensed and inactive form. And that lid is this globular receptor binding domain. So what needs to happen in order for this fusion machinery to be turned on and allow the virus membrane to mix with the cellular plasma membrane in order for the virus to penetrate that outer cellular defense of its plasma membrane, is that you've got to remove this lid to allow this fusion peptide to expand and access the cell. So that's the second really critical function about spike. I'm going to show you a movie about this in just a second. But first, you'll notice this is color-coded, and it's color-coded to show the degree of sameness or conservation across closely related spike proteins from members of the same subgenus. What you'll notice is that the areas in light blue or teal are the more variable region, and the darker areas in purple are the region that are less variable. And so, not surprising, for all viruses, it's true that the receptor binding domain, or almost all, is a region of the virus that is prone to intense evolutionary pressure, usually from immune system, from neutralizing antibodies. So this is a part of viruses that generally will change much more rapidly than many other regions of the virus. And that is true for coronaviruses as well. However, don't get the opinion that these are changing like crazy, because in fact, coronaviruses in the spike has overall pretty low genetic diversity, which means few new mutations tend to arise over multiple rounds of viral replication. And keep that thought in your mind, because we're going to touch later in this talk about why that's the case. But the fact that it's the case is important, because that means that this is probably good news from a vaccine strategy. We worry that vaccines that might be made, well, maybe the virus will mutate really quickly away from the neutralizing antibodies to escape them that are generated. And I think that that is less likely to happen for this coronavirus than for other viruses that have much, much higher mutation rates, things like HIV and influenza. It's not to say that there's no changes happening to the virus. Of course there are. And some of you may have heard about this D614G mutation that's been in the news a lot lately. And this refers to the fact that an aspartic acid at position 614, which is located sort of at the boundary between the receptor binding domain and the fusion machinery, pretty early on in the pandemic, I think in March or April, switched quite rapidly from being an aspartic acid to being a glycine. And a recent report shown here has shown that this change was perhaps something that was positively selected, meaning that this change may have benefited the virus in creating a variant of spike that was more transmissible or could access the interior of cells more easily. So there are these changes that are happening, and they're being documented through these widespread sequencing efforts. But the virus in general is not mutating at the rapid clip of some other RNA viruses. All right, so let's summarize what spike is doing to get the virus into the cell. First, as I said, it's got to act like a key to dock into the lock of that ACE2 receptor protein. So only cells that have this receptor have the capacity to allow the virus in through that interaction. But as I said, that's not enough. In addition, the second critical activity of spike is that it has to activate that fusion machinery on the sort of lower stalk region of the protein. And the removing or the opening of that lid happens by clipping it off using a sort of what you can think of as molecular scissors called protease enzyme. The protease that has been shown to play a major role in clipping off and activating that spike fusion machinery is called temporis-2, although it's thought that others might be able to be involved, like furin as well. So that protease clips off the tip of the head of the ACE2 receptor, and what this does is it allows that internal spring-loaded fusion machine to turn on. It inserts into the cellular plasma membrane, brings the viral membrane in close proximity with the cellular membrane, and fuses them. This creates a portal between the interior of the virus and the inside of the cell, so that the virus can now deposit its payload, which is that RNA genome, into the cytoplasm to start replicating. All right, so it's gotten in. Now what the virus needs to do is to convert that genome into a lot of proteins that are going to do the work of remodeling the host cell, copying that viral genome, and assembling new progeny virions. How does it do that? Well, let's first take a look at the viral genome itself. What is it made of? As I said, it's a long genome made of RNA. It encodes about 13 different genes. And so these are outlined in this diagram here. ORF stands for open reading frame. So it's a gene that is either made in a shorter form or a longer form. And you've got things like spike or S, envelope, matrix protein, other sorts of things. From these 13 genes, at least 27 proteins are made. And actually, a recent ribosome profiling report suggested that there may be an additional 20 protein products made through things like upstream open reading frames or other things like that. So how do you get, first of all, 27 or more proteins made from only 13 genes? Well, the answer to that is that the virus has to use some trickery. And it has to use some trickery for two different reasons. The first is that even though the virus can make use of cellular machinery, it's basically got to abide by the rules that those cellular machinery normally operate by. And in a eukaryotic cell, like our own cells, in general, we get one gene made into protein per messenger RNA. All right, so our messenger RNAs, which are the things that are read by the ribosomes, generally have a 5' cap that helps recruit the protein synthesis machinery, the ribosome, which will then scan through what's called untranslated region until it locates a start codon. This tells the protein synthesis machinery to start making protein, to start copying this gene from its RNA form into the protein form. And it will read through until it hits a molecular stop signal, and then the ribosome will leave. Everything downstream of that stop signal is not seen by the ribosome. So that's shown here, the ribosome, which will come, scan along, make protein that is within that blue region that I've shown here, and then dissociate and leave. This is a problem for a virus like coronavirus, right? Because I just told you it's got a whole series of genes. It doesn't have one gene on its RNA. It's got 13 genes on its RNA. But based on the eukaryotic rules of translation, these genes are going to be mostly not seen by the ribosome. The ribosome is going to come along. It will see gene 1. It'll find the start codon. It will translate that into protein until it hits the stop codon for gene 1. And then it will dissociate and fall off and then restart at gene 1, start again. All of these genes downstream or to the right of gene 1 are basically going to be invisible to the ribosome. So this entire RNA will not make protein 1, 2, 3, 4, and 5. It's only going to make protein 1. So how does the coronavirus get around that potential barrier to expressing its genes? Well, the first thing that it does is it uses a strategy in which it encodes many different proteins in a single gene. So it does this for this first giant gene that takes up a large portion of the viral genome, actually. And the way that it does it is that it inserts these genes into just this one open reading frame by removing those stop and start signals that are normally at the barrier, telling the ribosome when to begin and when to end. So it just has one start signal at the end of the first protein coding sequence, and then it has another one way at the end after the ribosome has gone through 11 in this case, or if it frame shifts here, up to 16 different genes can get made. This is called a polyprotein. It is essentially a giant fusion of between 11 and 16 different proteins made from one single open reading frame, or one single gene. Now, how these proteins then get separated from each other is through the use of those molecular scissors again, but this time not cellular molecular scissors. The virus encodes its own protease enzymes that can come and clip this long polyprotein into the individual protein subunits that will then each go and have different activities. As you can imagine, this protease is a really critical activity for the virus, and so it is a target for antiviral drug development, for sure. All right, so that's one strategy that the virus uses to get as many as 16 or more proteins made from one gene. But that doesn't solve the problem of all of these other genes that are now found at the right-hand end of the genome. How do those get made? Well, the virus uses a second strategy to get these sets of genes made into protein. And the way that it does that is by generating new RNA copies. And those new RNA copies are subgenome length. They're less than the full genome length, so we call them subgenomic RNAs. And they're a nested set, meaning that they all have the same 3' end, the same right-hand end here, that is basically the end of the viral genome. And they will sometimes, though, be long and have all of the genes present, and sometimes be shorter and only have some of the genes present. And so the way that this happens is that as the viral copying machine is making these RNAs, at the boundary between each of these genes found in the right-hand side of the genome is a signal. It's called a transcriptional regulatory sequence, or a TRS. And that signal basically is giving the RNA copying machine the option of stopping or going, and maybe like a stoplight. We don't know exactly how it works, but nonetheless, it allows that copying machine to sometimes stop at that signal, in which case you'd have a short RNA if it stopped at the very first signal, or sometimes read through that first signal until it gets to the second one. And then it does kind of a crazy thing, which is jump all the way to the other end of the genome and add this special 5' leader sequence, which is why these all have the same 5' end. That leader sequence may help with translation. It may help with other functions related to these RNAs. All right, so why does this solve the problem for the virus? Well, it solves the problem because by making these subgenomic RNAs, each of these downstream genes has the chance to be represented as the first gene that the ribosome protein machine makes or sees. So in this case, gene 2, which is akin to spike protein here, is the first gene that the ribosome sees. And so it will get translated into protein, and you'll have spike. All these other genes downstream, of course, won't get seen. But that's okay, because there's another RNA where gene 3 has the opportunity to be the first gene detected by the ribosome, and so on and so forth. And in this way, each of the proteins located on the right-hand end of the genome can get synthesized from these subgenomic RNAs. So it's really a combination of this giant polyprotein fusion and making subgenome length messages from the other end of the RNA that allows the virus to express all of its proteins, which are going to do the work of copying the viral RNA and remodeling the inside of the host cell. So let's talk a minute about this RNA copying machine. It's pretty fascinating. In fact, all RNA viruses have to encode their own RNA copying enzyme. This is called an RNA-dependent RNA polymerase, or an RDRP. And that's because our own cells don't have this machine in them for the viruses to steal. We've got protein-making machinery that they can steal, but we do not have RNA copying machinery. So every RNA virus has to bring in their own RNA copying machine. For coronaviruses, this RNA copying machine is remarkably sophisticated. It's got the core polymerase, shown here in pink as encoded by non-structural protein or NSP12. This is the core RNA-dependent RNA polymerase. But you'll notice that there's 8 or 9 different additional viral proteins that make up what's called this replicase complex. These are things that help the polymerase stick on the RNA and be more processive, that process the RNA as it's being made, that may unwind the RNA to get through troublesome structured regions potentially. But the complexity of this RNA copying machine is very high for coronaviruses. It actually rivals those of DNA replisomes, which are much more sophisticated in general than these RNA copying machines. Why might that be? Well, this is actually quite a fascinating facet of coronavirus biology. But for you to understand that, I need to remind you that, first of all, RNA copying machines generally make a lot of mistakes. Not on purpose, but they just, on average, will insert a wrong nucleotide every 1,000 or 10,000 bases that they're copying, all right? So this is what we call an error-prone polymerase, an error-prone nature of the polymerase. And it is what is driving the high rates of mutation that you see in many RNA viruses. Things like HIV have an astronomically high mutation rate because their RNA copying machine is inherently sloppy. Influenza, Ebola virus, all of these RNA copying machines are sloppy. They can't correct their errors, and that's what leads to high rates of mutation. Remember, though, that I said the coronavirus genome doesn't seem to be mutating at these high rates that we see for many other RNA viruses. The reason for that is this sophisticated RNA copying machine that the coronavirus has an ace in the hole. It's got its own proofreading mechanism built into it. So this is basically a copy editor that travels along with the polymerase. And if the polymerase makes an error, it puts the wrong nucleotide in the sequence. This copy editor will kick that error out and allow it to be replaced with the correct nucleotide for the sequence. So comparing this type of proofread genome, you could see that there would be many fewer errors compared to a viral genome that does not have this proofreading activity. This lower graph here are data from SARS coronavirus, indeed showing that if you eliminate this proofreading enzyme, which is called XON, the number of errors that randomly occur in the genome jump something like 20-fold. So this is likely the underlying reason why coronaviruses tend to have a lower error rate. And it also turns out to probably be a strong component, at least, to the reason as to why coronavirus genomes are so large. In fact, the coronavirus genome, which has about 30,000 different bases, nucleotides, is 2 to 3 times larger than an average RNA virus. Most RNA viruses are in the range of 8 to 18,000 base pairs. Things like rhinovirus, which is the cause of most of the common cold, is less than 10,000, as is HIV. Influenza is somewhat more than that. But you'll notice that SARS-CoV-2 and other coronaviruses, their genome is huge. In fact, they approach the theoretical limit for how large an RNA virus genome can be. And the thing that sets that limit is this error rate. So the problem with having an RNA genome, well, if it's a problem for the virus, and actually it usually isn't, but the issue of genome size is that the longer the genome that you have, the more chances that as that RNA copying machine comes through, it will randomly make an error, right? So if you make an error once every 10 10,000 bases, and if you have a genome that's 10,000 bases long, well, you're going to have an error randomly somewhere every time you copy that RNA. The longer, but if you've got a longer genome, then you're going to have more than one error every time you copy that RNA. And it turns out viruses can tolerate a certain number of random errors. But if there are too many, this decreases what we would call the fitness of the viral population. And they undergo what's called error catastrophe. Too many errors, it makes the virus too weak, too many problems, and the whole population can crash. It's this that helps determine the limit of how long a virus can be. So the fact that the coronavirus can correct its errors enables it to have a much larger genome before it might hit this error catastrophe threshold. And that means that the coronavirus genome can encode more genes, more proteins to remodel and have perhaps more sophisticated interaction with the host cell. All right, so now that the virus has made its set of viral machinery, what is it going to do with those machinery? How does it use each of those viral proteins to reorganize the host cell and start making lots of new progeny virions? Well, it turns out that it really strikingly remodels the inside of an infected cell. And it does this by essentially creating new rooms where the virus can replicate. And by new rooms, what I mean is that there are viral proteins that are made that act on cellular membranes derived from the endoplasmic reticulum, the rough endoplasmic reticulum, that take this cellular membrane, move it and stretch it, and use it to form these circular double membraned vesicles that form throughout the cytoplasm of a coronavirus infected cell. These are called replication and transcription complexes, or RTCs, but essentially what they are, is you can think of them, are separate rooms within the cell that allow the virus to copy its genome really efficiently. So there are probably two reasons, at least two reasons, why these rooms are created by the virus. One is that it helps the virus do what it's doing, which is copying its genome in a more secret location. The cytoplasm of the cell is full of sensor proteins that are looking for evidence that the cell has been infected. It's looking for things that look foreign, because if it detects those foreign things, it's going to alert the broader immune system to try and kickstart the adaptive immune response to produce antibodies and killer T cells, and also to warn all of the neighboring cells to mount their immune defenses to protect against infection. And a prime signal that these sensor proteins look for are double-stranded RNAs, because these generally don't form within our own cells very effectively. But if you think about how an RNA genome is copied, you're starting with one incoming strand of RNA, and the RNA copying machine has to make a copy of that. And so that copy can form double-stranded RNA intermediates, which are a key signal to the immune system that they're infected. So if you create specialized environments where these RNA copying reactions are happening, there may be less of a chance that the immune system can detect them. A second hypothesis for why these are beneficial is that it allows the virus to basically concentrate all the things that it needs to copy its genome and to transcribe its genome in one place. And so the whole process may be more efficient. These are the rough endoplasmic reticulums, so sort of studded with ribosomes around the periphery of these, allowing new messenger RNAs that are made from the viral genome, perhaps to be translated into protein in the same general location as well. So this is an image taken from a SARS coronavirus infected cell, just to give you a sense of these double membrane vesicles are really filling the cytoplasm of these coronavirus infected cells. They're pretty much everywhere. These circles are these vesicles that the virus is forming. And a zoom out here, this is cryoelectron tomography experiment, and this is a 3D rendering of that. Basically allows you to build a three-dimensional structure to see what the inside of a cell looks like. Allows you to see this is pseudo-colored, that these vesicles, the inside of these vesicles would be this area here that is surrounded by the purple, and then because they're double membraned, you have this second membrane that is yellow, that is contiguous, that sort of loops around the vesicles. And so inside each of these circular areas would be where viral RNA copying is taking place. In addition to remodeling the membrane environment of the cytoplasm, the virus is also pretty dramatically changing the gene expression environment of host genes. And one of the ways that it is doing this is through a protein that is the first protein made off of the viral genome. It's called nonstructural protein 1. This is a viral protein that is able to potently restrict cellular gene expression. And it does this through a two-pronged strategy of both inducing cleavage of messenger RNAs through a mechanism that is not yet well understood, and through repressing translation of the messenger RNAs. And this is something that for the SARS-CoV-2 NSP1 protein, there have been a series of recent studies and preprints that have really helped elucidate the mechanism of how this protein restricts translation. So the 40S subunit of the ribosome is one of the major components of the ribosome, of course, that helps translate messages into protein. And the way that it does this is it's got an entry channel that allows the messenger RNA to feed into the ribosome so that it can be read by the ribosome and converted into protein. Turns out what NSP1 does is it's got this unstructured domain that is highlighted in pink here that inserts into that mRNA entry channel of the 40S ribosome like a cork, essentially preventing the messenger RNA from accessing the ribosome and thereby reducing translation of these messages. So why might it be beneficial to reduce translation of cellular messages? One hypothesis is that it could shunt available ribosomes preferentially towards viral messages, for example, if they were more efficiently translated. Another hypothesis that's been widely discussed is that this could be a very good general immune evasion strategy for the virus. So remember those sensor proteins that I was telling you about in the cell that are constantly surveying the cellular environment for evidence of infection. And when they detect that evidence of infection, they're going to then use that information to convey a signal to the nucleus that tells the cell to turn on genes that could be involved in fighting infection. Genes like those that encode interferon, which is a sort of a major SOS signal, or interferon stimulated genes, or a whole suite of things that cells can use to activate the innate immune response. Well, the thing about needing to turn on these genes is that the cell has to transcribe them into its own messenger RNA. Those messages have to be read by the ribosome in order to produce things like interferon. So if NSP1 comes along and blocks this message from being read by the ribosome, then the cell will be able to produce less of the signal that activates the innate immune response. And that's shown here from this preprint by Thoms et al., showing that under conditions where you activate the innate immune response strongly, this is through actually infection with another RNA virus called Sendai virus, these cells will secrete a lot of interferon beta, which is one of these molecules that activates the immune response. However, if the cells are expressing the NSP1 protein, much less interferon beta will get produced because the interferon beta messenger RNA can't be effectively translated by the ribosome. So this fits in with some observations from transcriptomic studies that were published by Dentenover's group, showing that in SARS-CoV infected cells at low MOI, these viruses tend to induce, or the infected cells tend to have a pretty limited innate antiviral state. low expression of interferon-stimulated gene products like interferon beta and interferon lambda. So this NSP1 activity could be contributing to that. There are probably a suite of other proteins that contribute to that too. I'll point out that in the coronavirus genome, in addition to the enzymes that are copying the RNA and the structural proteins that are making up the viral particle, there are a whole suite of what are called accessory genes. These are genes that are generally unique to each virus and are thought to fine-tune the interaction between that virus and the host environment in vivo. And so while we don't know the function of many of the accessory genes for CoV-2 yet, the hypothesis is that based on what we know from related coronaviruses, that they're likely going to be involved in modulating the immune response as well, and probably contributing to this phenotype. All right, so now the last thing that the virus needs to do basically is take those copied genomes that it's made and assemble them into new progeny virions that can then be released from the cell. And this is a process that's, again, sort of largely mysterious for coronaviruses, and in part because I told you that the RNAs themselves are made in these protective vesicles. Somehow they need to get transported out of those vesicles and coated with the nucleoprotein and directed to a new set of vesicles that are derived from the ER Golgi intermediate compartment, or the ERGIC compartment. These vesicles have been pre-studded with the spike protein, the matrix protein, the envelope protein that are going to form the outside of that new coronavirus particle. And so interactions between the nucleoprotein coded genome and probably the matrix protein drive a budding reaction that causes the viral genome to be enveloped within these vesicles to form these new particles. So it looks something like this, where the genome would be taken in and enveloped and then pinched off. So these vesicles would now have a series of new virus particles within them that are then transported through an exocytosis-like mechanism to the surface of the cell, where they're released to go on and either infect neighboring cells or be breathed out into the environment to infect a new host. There's a recent paper that just came out showing that CoV-2 infected ViroE6 cells in this scanning electron microscopy image. You can see that the viral particles, which are these little dots here or here, seem to be being released from these large protrusions from the infected cell. These are called filopodia, and they're filled with a molecule called actin. They're actin-rich. Actin is a component of the cellular cytoskeleton. It helps form the structure of the cell, but it is also important for cell movement and things. And we know that other viruses like vaccinia, which is a member of the pox virus family, or Ebola, or others, are known to dramatically remodel actin or use actin to help them escape from cells. So a hypothesis here is that CoV-2 is also using actin to help create or hijack these filopodia to push the viral particles away from a cell that they're produced in towards the extracellular environment or towards a neighboring cell. And so that's going to be an exciting area of future research. All right, let me summarize what I have covered today, which is basically the entire coronavirus life cycle inside a cell. We discussed how the first thing that happens after the virus is inside the body is that it has to locate cells that have the right lock, the right ACE2 receptor, and that spike protein binds that receptor in a lock and key-like mechanism, but then uses a second activity, its spring-loaded fusion machine, to enter past the cell's plasma membrane defense, to get in through the door. It opens the door by fusing the viral membrane with the host plasma membrane, depositing its RNA genome into the cell. It then uses some viral tricks to get 27 or more different viral proteins translated from the information on this single incoming RNA And that's through a combination of a giant polyprotein fusion that's processed and by making smaller subgenomic RNAs. These viral proteins then do the work of reorganizing inside of the cytoplasm of the infected cell, stretching cellular membranes to form these new rooms in the cytoplasm, these double membrane vesicles, where the RNA copying machine, which has proofreading activity, is able to then make lots of copies of the viral genome and make subgenomic RNAs to make more viral proteins. These proteins, the structural proteins, are then embedded in new vesicles in the cytoplasm, and the newly copied RNA is transferred to those vesicles coated with nucleoprotein and buds into them to form progeny virions before they're released from the cell. So in this way, the virus can start with a single incoming virion, a single RNA, and release hundreds of progeny virions from that single infected cell in a remarkably short time frame, 8 to 10 hours for the average coronavirus infection. All right, I'm going to end there and just acknowledge that I had a lot of assistance for image preparation as I was putting together this and other coronavirus-related talks from my lab. Ella Hartinian, Divya Nandakumar, Jessica Tucker, Michael Lee, and Azra Laurie all helped. We often use BioRender to make these images. I also had a wonderful collaboration with Scientific American to create several of the images in the movies that you saw with the editor Mark Fischetti and the illustrator Veronica Hayes. And so you can see those movies in this interactive website here. And I also want to acknowledge that the data that I was showing today is really not from my group. It's from a whole collection of coronavirus researchers and scientists and clinicians who have become coronavirologists in order to generate this type of information so we can learn about how this virus functions and attempt to combat the pandemic. So with that, I hope I've left enough time to take some questions.
Thank you very much, Professor Glaunsinger. There are a bunch of questions. Charles wants to know, are the matrix, E, and spike proteins the only proteins found on the coronavirus envelope?
Yeah, as far as I know, those are the only viral proteins that are there. It's entirely possible that some cellular proteins come along for the ride, but if there are cellular proteins there, we don't know of any functions that they may have, and it could just be that they happen to be on that membrane when the virus is spreading. So usually we'll identify proteins that are there by collecting viruses and subjecting them to things like mass spectrometry. So it's possible there are some others, but these are the only functional ones I'm aware of.
And Jarek's asking, can the virus selectively repress translation of cellular genes without repressing translation of its own RNAs?
Yeah, this is an excellent question, and something that is, there's some mixed information on that, I would say, in the literature. Conceptually, it would make the most sense for viral RNAs to escape this translational repression. And it's not clear that they do entirely. But let me tell you the thinking behind this. So one hypothesis is that even if viral RNAs could be subject to this repression, it turns out that from related coronavirus studies where they're looking at the composition of these membrane vesicles where the RNA is being produced, the NSP1 protein is not found there. So it's possible that it's located during infection elsewhere in the cytoplasm in areas where cellular RNAs are generally being translated, but not in close proximity to where the viral RNAs are being translated. So it could be that there's a sort of a separation of location that helps it selectively dampen host gene expression. There are also some data that suggest that the viral leader sequence that gets appended to that end of the viral RNAs may confer a translation advantage to those RNAs. This has primarily been shown in vitro studies, and recent ribosome profiling study did not detect that translation advantage. So I think the jury is still out on to what extent that happens. And then there's the idea that coupled with perhaps if the viral RNAs do have a translation advantage, then by selectively depleting some of the ribosomes or inactivating some of the ribosomes, you could nonetheless shunt the rest of them. They would be preferentially occupied by viral RNAs instead. So it's a complicated answer, but a great question.
One of the students wants to know if the virus encodes its own protease, won't the viral protease be part of the polyprotein? And if so, how can it splice itself?
Yeah, another excellent question. It is part of the polyproteins. The viral proteases, there are two of them actually, are part of the polyprotein, and they function both in cis and in trans. And so what that means is that the protease, as soon as it's translated by the ribosome and that domain of the protein folds, it can become active and self-cleave out of the polyprotein. So it can cleave itself out and then go and act on the rest of the junctions to cleave those polyproteins into individual unit proteins. So you're correct, it's in the polyprotein, but it can cleave itself out of the polyprotein and act on the other junctions.
And Arbery's asking, does the ACE2 receptor vary in abundance among the human population? And is it also found in bats and pangolins?
It is not restricted to the human population, so ACE2 can be found elsewhere. Whether it varies in abundance in the human population, it's an interesting question. I don't know the answer to that. But I did see a nice study by Ralph Barrick's group a few weeks ago, which studied the abundance of ACE2 receptor in lung tissue. And what they found was that the highest concentration of ACE2 was in the upper respiratory tract, where most people are getting this coronavirus infection. And the amount where the concentration of ACE2 sort of decreased in a gradient the lower you got in the lung, and so that the deep lung had less, which could potentially explain why not everybody, thankfully, is getting this fulminant deep lung infection, which is the most dangerous form, and that it is more often restricted to the upper respiratory tract. So the concentration of ACE2 in different tissues or different locations within the body certainly can vary.
And Ari'd like to know, how do we know how the viral genomes are successfully coded in the nucleocapsid? Why doesn't the nucleocapsid encapsulate host cell genomes, host cell RNAs?
Yep, another answer that we don't know why. It is probably a feature of location, that the nucleocapsid, well, first of all, there may be fewer cellular RNAs around because they've been depleted through the process of host shutoff. And second, nucleocapsid is being, it is within those compartments as well, those replication compartments. So it's quite possible that it is coding the viral RNA there, and together they are escaping and going to the location where viral morphogenesis or budding is happening. I think it's entirely possible that nucleocapsid could bind cellular RNAs. Nucleocapsid is the most abundant viral protein that is made in these cells, and not surprising because you need a lot of it to coat all of the viral RNAs that are made. And it plays additional regulatory roles that I didn't talk about beyond the morphogenesis component. But my hypothesis would be that it's because it is made perhaps in proximity or is located in proximity to where those viral RNAs are amplifying, it will preferentially code those.
Another student wants to know if there's a mutation in the proofreading enzyme that inactivates it, wouldn't this be beneficial for CoV-2 and allow it to more rapidly evolve?
Yeah, that's kind of a fascinating concept. And to some extent, the answer could be yes, but as it turns out, most RNA viruses are thought to have sort of hit at the sweet spot for the maximal number of mutations that they can tolerate without going overboard. As you're alluding to, some mutation does benefit the virus, right? This allows for inherent flexibility in the virus to encounter, if it encounters a bottleneck somewhere, if it encounters an immune pressure somewhere, well, there might be some variant within the viral population that's arisen through this random mutational event that allows that virus to bypass the error. And so there are some beautiful early studies actually with poliovirus from Carla Kierkegaard's lab and Raul Andino's lab showing that indeed, this is truly beneficial for the virus to have some level of mutation in an in vivo setting. But it's a double-edged sword because too many mutants, the virus cannot tolerate. And so many of the antiviral drugs that are used against viruses, or sort of the best studied ones, these target the polymerase. And several of them are of the class where they increase the error rate of the RNA copying machine, maybe just 2 or 3-fold, but that's enough to push that virus towards what I described as error catastrophe. They cannot tolerate more mutations and still have a fit population. So it's a very fine-tuned rate of error probably for each virus.
Britt, thank you very much on behalf of the students. It's been very illuminating to hear you tell us about this virus. Thanks for spending time with us.
My pleasure. Thank you for having me.